Hi, welcome to this next video tutorial in your General Biology course. This session is entitled Energy and Metabolism Part 2. So in the previous video, we talked quite a bit about energy, the laws of thermodynamics, and how energy is needed to drive a lot of metabolic processes. In this session, we want to talk about how enzymes are necessary to catalyze or speed up those metabolic reactions. So let's take a look at our learning objectives. First, we'll need to talk about activation energy. We've talked about Gibbs free energy, and this plays into that just a little bit, but we'll need to see what activation energy is. Then we'll need to talk about enzyme structure and function, see what they're made of and how they work. We'll need to talk about enzyme regulation as well, because that's ultimately how we control our metabolic processes. So let's remind ourselves of the 6 features of life that we've been talking about. We've talked quite a bit about energy processing, but we need to finish that discussion up by discussing enzymes because they really lead us into that step of regulation that you see on the list and help explain how cells regulate metabolic processes. So again, in our last video, we defined metabolism. We said it was the sum of all biochemical reactions that are occurring in a cell. And we said there's 2 types of metabolic reactions, catabolic and anabolic. Catabolic reactions are decomposition, taking things apart, so Z breaking down into X and Y. So we're breaking bonds, and when you break bonds, you're releasing energy. And the name for that is exergonic. So exergonic reactions release energy. And then there are anabolic reactions, which we said were synthetic, so you're synthesizing, building, putting together, like A plus B are combining to form C. That means we're building bonds and we have to supply the energy to do that. And those reactions are called endergonic, when you have to supply energy to do them. And we said graphically, exergonic reactions are going downhill. So we said there's more energy in Z than there is in X plus Y. So we're going down, we're losing energy, and that's a negative delta G, a negative change in free energy to go down. And compare that to the endergonic reactions, which are energetically uphill. A plus B forms C, but we had to provide that energy to get up there. And so that's a positive change in free energy, or a positive delta G. And then we said that living things have figured out how to do energy coupling. They have figured out how to couple an exergonic process to an endergonic process, and that's ultimately what allows life to do such complicated processing and build structure and order that seem to be defying the laws of thermodynamics. They're not really defying the laws, it's just interesting to see life build complexity and order. But notice here that the exergonic process is releasing more energy than the endergonic one requires, so there's still an excess of energy being released. The delta G for the combined process was minus 60. And because the delta G is negative, that's exergonic. And recall that another term we could be using here is spontaneous. If a reaction is energetically downhill, if it has a negative delta G, we could say exergonic, but we could also say that it is spontaneous, meaning that it will happen. The laws of physics, the laws of thermodynamics are in our favor, so spontaneous means it will happen. We don't have to force it. But we need to ask the question, is this reaction instantaneous? Because spontaneous and instantaneous do not mean the same things. Spontaneous simply means that we're energetically favorable, we're ultimately energetically downhill. Instantaneous is asking a question about the speed or the rate at which the process unfolds. So not all spontaneous reactions are instantaneous. However, in cells, we need reactions to be essentially instantaneous. Cells need reactions to happen extremely quickly, like a matter of nanoseconds. So let's talk about how we would know if a reaction is instantaneous or not. So here is a graph that's plotting the change in energy as we go from reactants to products. And as you can see in this graph, the amount of energy that's invested in the reactions is up here, and the amount of energy in the product is down here. So we went down. That's ultimately a negative change in energy. So we know that this reaction is exergonic. We also know, therefore, this reaction is spontaneous. A will break down into B if we just kind of let things run their course. But again, we need to think about the speed or the rate. And to understand that, we need to look at this change in energy right here. Now, this doesn't affect whether the overall process is endergonic or exergonic. If you want to know if a reaction is endergonic or exergonic, you simply ask, where did I start, where did I finish? And we started up there where A is, we ended down there where B is, so it doesn't matter the twists and turns of the graph. Where did you start, where did you finish? We went from higher to lower, so this is exergonic. But this little hill right there, this energy bump, E sub A, that is the activation energy. This is the energy that's required to initiate the reaction. So notice it's kind of like a roller coaster. We've got to invest some energy to get the reaction up to the top of the hill, and then we can ride the roller coaster back down and more. So ultimately exergonic, but there is an investment of energy required up front, and that's the activation energy. And the height of that hump, the height of that activation energy curve there influences the rate or the speed of the reaction. So the higher that bump, the higher the activation energy, the slower the reaction. Whereas if that roller coaster hill at the beginning was lower, that would mean a faster overall process. So the height of or the amount, I should say, I guess, of the activation energy requirement determines the rate. The more energy required, the slower the reaction takes place, and the less energy required, the faster the reaction takes place. So in other words, if we want to speed up this reaction, we're going to have to provide that activation energy, or that's one way to do it, is to provide it. And that's what we could do with heat. We could simply heat up the reaction and give the reaction the activation energy that it needs. So when you heat things up, you make the molecules move faster and reactions proceed faster. So using heat is simply to supply the activation energy. But is this the way cells would want to do it? And the answer is no, because heat could actually cause a lot of problems in the cell. First of all, heat would possibly denature proteins and other things, creating trouble. But the other real reason that cells don't use heat is that heat is a non-specific catalyst. By the way, to catalyze means to speed up. So it is true that heat would catalyze or speed up the reaction, but it is non-specific. So what do I mean by that? What I mean is that heat would speed up A breaking down into B, but heat would also speed up every other possible process in the cell. There would be no way to tell the heat which reactions to speed up. And cells have to be very particular about the reactions they're catalyzing, because sometimes they want to be making this substance, sometimes they want to be breaking this substance down, other times they want to make this but not make this other thing. So they have to pick and choose what processes are taking place, and so we can't just catalyze everything. We need a way to catalyze specific chemical reactions at specific moments within the cell. And the way cells have figured out how to do that is to use enzymes. So let's talk about enzymes. Enzymes are proteins. So this is one of the roles that proteins serve in a cell. Enzymes are proteins with unique three-dimensional shapes that catalyze specific biochemical reactions by lowering the activation energy requirement. So notice I didn't say by providing the activation energy, which is what heat does, rather by lowering the activation energy requirement, making it so the cell doesn't need as much activation energy to perform the process. And by lowering the requirement, that's synonymous with speeding up the reaction. But it also catalyzes specific ones, and that has to do with the three-dimensional shape. So in this picture, we see an enzyme, which is a protein folded into a shape, and the reactant comes in, and it's going to bind to this active site. So enzymes have a little folded pocket or groove up there. You see the little indentation we're calling the active site. It is this site that's going to perform the catalysis or help speed up the process. So what binds to the active site? Well, that would be something called the substrate, which you might also think of as the reactant. So this is the substance we're starting with, this substrate. And in this particular example, we're going to use this enzyme to break the substrate down. So the substrate is going to come in and bind to the active site, and notice that the active site has a shape that fits the substrate. It's hard to tell here because the substrate's off to the left, but in the next picture you're going to see that that substrate will fit into that active site, almost like a lock and a key, or some people use hand and glove. There's a lot of metaphors for this. But let's see how that happens. So here's our enzyme, and here comes the substrate, and it's going to bind to the active site. And once it's in the active site, the enzyme is going to put some strain on those bonds, and that is going to help break this thing down, and so the product will be released. In this case, we're catalyzing a catabolic reaction. We're breaking that substrate down into two simpler substances. So this is a catabolic reaction that's being catalyzed. And once the product is spit out, the enzyme is good as new. The enzyme itself hasn't been changed at all. So the enzyme is free to come back here and accept some more substrate. So this enzyme could continuously be catalyzing the breakdown of that substrate. Now again, the example I'm showing you here is a catabolic process, but enzymes are also used to catalyze anabolic processes. Now it'd be a different shaped enzyme, but you could have an active site that could bring in two reactants and help forge them together or bond them together to form a new product. So enzymes are used for anabolic and catabolic processes, but we need a unique shaped enzyme for each particular reaction. So let's look graphically at what took place there. So here is the graph we saw just a minute ago, and you see that activation energy curve or bump. And remember, the higher that hump, the slower the process. And the whole goal here is to speed up the process. So this is what happens without the enzyme present, but let's look at the same graph when there is an enzyme present to help catalyze the breakdown of A into B. So notice that the activation energy requirement is less. We've lowered the curve, and that is synonymous with speeding up the reaction. So again, heat would be providing the activation energy, whereas enzymes are lowering the activation energy requirement, synonymous with speeding it up. And what allows an enzyme to catalyze a specific chemical reaction is that three-dimensional shape. You've got to have the right active site, the right little lock and key groove and pocket there to fit the substrate. And the reason that enzymes can have unique shapes is because, again, they are proteins. And we have said that protein shape determines protein function. We've said that in some previous videos. And so proteins are amino acid chains that have folded up. And if you recall, we talked about primary, secondary, tertiary, and quaternary structure. So ultimately, an enzyme is a chain of amino acids, that's its primary structure, that's folded into secondary and tertiary structures, and sometimes quaternary, depending upon our example. Now, because enzymes, being proteins, are so dependent upon their shape to do their job, they are also vulnerable to being denatured and then not being able to do their job. So maybe here's an example of a folded up protein. It has its three-dimensional shape, and somewhere in there you would note an active site, the little pocket or groove where substrate binds. But recall that protein folding is dependent upon temperature, pH, and salt concentration. The reason the folding is dependent upon those is because those three variables, temperature, pH, and salt concentration, can affect hydrogen bonds, where and how many hydrogen bonds are forming. So if we mess with the temperature, the pH, or the salt levels, we can essentially risk denaturing the protein by disrupting some of the hydrogen bonds that were holding it into its shape. And that has implications for enzymes. Your cells have got enzymes that need to be folded correctly, and that's why it's so important to maintain a constant body temperature, a constant pH, constant salt concentration levels, like with the isotonic conditions. And we used a word earlier, we said homeostasis. Cells need to always be in a state of homeostasis so that these variables can be held constant, their proteins fold correctly, and their enzymes work correctly. Because if the enzymes become denatured, they're not going to be able to function. In fact, we can see that graphically here. So in this curve, on the y-axis, we're plotting enzyme activity. In other words, the ability of the enzyme to do its job. And you see the arrow going up. So another way to think of that is maybe how much product is being made. That would tell us how well the enzyme is doing its job. So that's what we're plotting on the y-axis, the ability of the enzyme to function. And then on the x-axis, we're plotting a condition, in this case temperature, and we're trying to see how does temperature affect the enzyme's ability to function. And we get this graph that's generally called a bell curve. And in this bell curve, what you see is that there is an optimum temperature at which the enzyme is working the best. And if this were an enzyme in your body, your human body temperature is 37 degrees Celsius. So we would say this enzyme is folding correctly at 37 degrees Celsius. We've got an active site. So the enzyme is doing its job very well. But if we strayed away from that temperature, you see as we get hotter, or for that matter, as we get cooler, the graph is dropping off. And the reason the enzyme isn't working as well as it gets hotter is it starts to denature and fold differently. It loses its active site and it's no longer able to catalyze reactions. Now, interestingly, we get a very similar curve if we were to plot enzyme activity as a function of pH. So pH also affects the folding pattern of proteins, including enzymes. And again, we see this bell curve. So that means there is an optimum pH where the enzyme works best. And again, if this is in your body, human body pH is approximately 7.35, right around the neutral value. And so that's where the enzyme is folding correctly, is at this neutral pH. And if you got too acidic or too basic as you stray from that optimum, once again, the enzyme is going to denature and fold differently and lose its active site. And once the active site has been lost, there's really no way for the enzyme to catalyze a process. This also explains, for example, why we cook food. When we cook food, we're denaturing any enzymes that might be present in the food, specifically the enzymes of any bacteria that might be present in the food. So by denaturing those bacterial proteins, we assure that we kill the bacteria off and makes the food safe to eat. With the pH curve, this is the reason why we clean things with acids or, like vinegar, or maybe with a strong base like ammonia, because by wiping things down with strong acids or bases, we will also be denaturing proteins in those areas, including enzymes, again, killing bacteria, but also destroying things like viruses that are made of protein as well. All right, so we see now how enzymes catalyze biochemical reactions. They have an active site, that little nestled pocket or groove there that the substrate comes in and binds to, and then the enzyme does its catalytic work and kicks out the product. So we see how that works. But now we need to think about how can enzymes be regulated? How can we control how much product is made? Maybe we don't want the enzyme just continuously cranking out product because we would end up with too much product. So how can we regulate enzyme activity? Well, again, let's think about the active site. That's the catalytic portion where the substrate binds to the enzyme. So we've discussed that. But there is another site on this enzyme called the allosteric site, and we hadn't mentioned that yet. Allosteric means change of shape. So this is a site where something can bind and alter the shape of the enzyme. So the allosteric site is going to allow for enzyme activity to be regulated by something called negative feedback inhibition. So again, if something binds to the allosteric site, the change of shape site, it won't catalyze something. What will happen is the enzyme's shape is going to change. And if the shape of the enzyme changes, so will the shape of the active site. And this is all going to work by something called negative feedback inhibition. So let's see what that is. Negative feedback inhibition occurs when the product of a process inhibits the process by which it was made. So that's negative feedback, when the product has a negative effect or a negative impact on its own production when the product turns off production of the product. A good example of negative feedback inhibition would be your air conditioner in your house. So the job of the air conditioner is to crank out cold air. The product of the air conditioner is cold air. But eventually it is the cold air that's going to come back and turn off the air conditioner. That's what the thermostat is for. So the product of the air conditioner, the cold air, is ultimately what will turn off the air conditioner when enough cold air has been produced. So that's negative feedback inhibition. But what does that look like in cells with a metabolic process? Well, a lot of chemical processes in cells have multiple steps before you get to the product. And each one of those steps is going to require a different shaped enzyme to get us to the next stage of the process. And that's where we often see negative feedback inhibition. So here's an example. Here we have an enzyme, and you can see substrate is getting ready to bind to the active site. And so that enzyme's job is going to be to convert the substrate into some other substance. So the substrate comes in and binds, and then the enzyme kicks out intermediate substance A. That's the initial product, not the final product, but the initial product. Intermediate substance A is slightly different in shape than the original substrate. But recall, we've also got this allosteric site on the enzyme. Nothing's happening there yet, but we're going to see something there in just a minute. So this first enzyme in this chain of events has produced intermediate substance A. Well, intermediate substance A is now going to be the substrate for another enzyme. So enzyme 2 enters the story. This is not the same as enzyme 1. It's got a slightly different shaped active site because intermediate A is not the same shape as the original substrate. So the intermediate A binds to this enzyme, and that cranks out intermediate B, kind of our next initial product but not final product. So we've gotten through 2 steps here, and we've got intermediate B to work with. Well, intermediate B now binds to the active site of yet a third enzyme with a different folded up shape, so it has the right active site for this. And once intermediate B binds to that active site, we finally crank out our final end product, that little red circular looking substance. So this is a multi-step pathway that was trying to generate this final end product. But the way we're going to regulate the amount of end product produced is through negative feedback. And notice that the end product right there is exactly the right shape to bind to the allosteric site in enzyme 1. So as product begins to accumulate, it comes back up here and binds to the allosteric site of that first enzyme. And again, allosteric means change in shape. So notice that when the end product binds to the allosteric site, the shape of the enzyme changes. It causes that protein to fold differently, and in fact, we've lost our active site. Notice that this substrate does not fit in that active site anymore. In other words, the enzyme has been turned off. So we're no longer going to get intermediate A, which means we'll not get B, which means we'll not get end product. So we're sort of nipping the process in the bud. When we've got enough final product, it comes back and turns off the very enzyme that's needed in the pathway to produce that end product. Now, as that end product, those little red substances, those little round red substances get used or altered in some way in the cell, then they will no longer be present to bind to the allosteric site, and the enzyme will go back to its correct shape, and it's like we've turned the process back on. And so this is going to allow us to regulate the amount of product. It's very self-regulating. When lots of product is present, it shuts down its own production, and as the product concentration starts to decrease, we see that first enzyme getting turned back on and more product being made. But as more product gets made, it will shut the first enzyme off again. So the amount of product is going to remain very constant. And there are a lot of substances in our cells that we need to maintain a very constant amount of. And negative feedback inhibition is the perfect way to regulate an enzymatic process so that the product remains at a very constant level within the cell. But sometimes we don't want the amount of a product to simply remain constant. There are certain substances that enzymes produce that we want their concentration to fluctuate, to go up and down, to be a lot of it for a time being, and then be almost none. So how can we make an enzyme produce a lot of product and then just say stop making that product and then start making it again? How can we create a scenario where we could turn the enzyme on and off at will, almost like a light switch? Well, one way to do that is through enzyme phosphorylation. So let's see how this works. There are some enzymes that exist in your cell already in a turned off state. So this is an inactive enzyme. So here's an enzyme sitting around in the cell that has no active site. So even though the protein has been produced, it's not folded into a shape that gives it an active site. So the enzyme is present but inactive, cannot catalyze anything. But we can turn it on using the process of phosphorylation. Phosphorylation simply means adding a phosphate group to it. So recall the phosphate group. That was one of the functional groups that we talked about in an earlier video. And if you recall, I said the important thing about the phosphate group is that it's negatively charged, and that makes it very hydrophilic and polar. And when you introduce these negative charges to that protein, those negative charges are going to impact how the protein folds. The negative charges are going to exert an effect on the amino acids in that folded up chain, causing it to fold differently. So we could say the binding of phosphate to inactive enzyme causes the enzyme to fold into an active shape. So again, we pop the phosphate group on there, and the next thing you know, the protein is folding completely differently, and now we have an active site. So now the enzyme is active. We've turned the enzyme on. Substrate can come in and bind. We can start cranking out product. So we let the enzyme crank out product and let the amount of product increase in the cell, and then we say, okay, that's enough. We want to turn the enzyme back off. Well, the way you do that is simply pull the phosphate group off. This reaction is reversible. So if we remove the phosphate, then we go right back to having an inactive enzyme. And so this phosphorylation, this adding of a phosphate group, is reversible. And the reversible binding of the phosphate allows the enzyme to be turned on and off like a lamp or like a light switch. So the enzyme is already in the cell, just inactive. When we want the enzyme to crank out product, we pop a phosphate group on there. That changes the folding pattern. The enzyme's now turned on. Substrate can come in and bind, crank out product, and then when we're done, just pop the phosphate off, and it's just like turning your appliance off. We go back to that inactive state. Remember, negative feedback inhibition keeps the amount of product constant at all times, whereas enzyme phosphorylation allows us to regulate the amount of product quite a bit. So you can have a lot of product or almost no product. Again, it's like turning a light on and off. Your refrigerator is an appliance that you want to have on all the time, so it's constantly keeping a kind of a baseline level of coldness. But then a light is something you only want on at certain times, other times you want it off. So that would be a case where we would do something like enzyme phosphorylation. Now there are a couple of other ways to regulate enzyme activity. Another way to do it is by competitive inhibition. So let's see what that is. Here's the enzyme, and you can see the substrate that comes in and binds to the active site. And when the substrate comes in and binds, the enzyme will crank out the product. So how can we inhibit this? Well, what if we have a substance that is not the substrate, but has a similar enough chemical structure to still bind in the active site? Well, then that could come in and block the active site, preventing substrate from getting in there. So a competitive inhibitor binds to the active site and blocks the substrate from binding, thus inhibiting enzyme activity. If we want to see that in a picture, it would look something like this. So notice when this yellow substance, the competitive inhibitor, comes in, it binds to the active site, And while it's sitting there, it is not possible for the substrate to bind, and as a result, there is no product synthesized. The inhibitor is just sitting there. The inhibitor is not reacting or being changed. It's just sitting there blocking the substrate. So a competitive inhibitor can inhibit or shut down the enzyme. Now, eventually, the inhibitor itself will probably come dislodged and move out, and then we'd kind of race again. Who gets in there first, the inhibitor or the substrate? So it's not a permanent binding that's going on there. It's still temporary. But while the inhibitor is sitting there in the active site, the enzyme can't crank out any product. Now, interesting, there are a lot of drugs that we prescribe because they are competitive inhibitors. Maybe there's an enzyme in the body cranking out too much product, and we need to slow that down. And so scientists and researchers have created competitive inhibitors to go in and block enzymatic function. And an interesting example of this is the drug Viagra. So there is an enzyme in our bodies that prevents vasodilation. And vasodilation is necessary for a male to have an erection. We need vasodilation for that to occur. It increases the amount of blood flow to the area. But there are enzymes that prevent vasodilation, and so Viagra's job is to inhibit those enzymes. If we can shut down the enzyme that's inhibiting the blood flow, then you can have increased blood flow to the area, and that's how the Viagra pill actually works. So it is a competitive inhibitor of an enzyme. Now, another interesting way to block enzyme activity is through non-competitive inhibition. So let's see what that's about. So there's our enzyme with the substrate binding to it, but non-competitive inhibitors bind to the allosteric site on enzymes and alter the enzyme shape, thus inhibiting enzyme activity. So recall, there's the allosteric site, allosteric meaning change in shape. We'd already talked about how a product can bind there and exert negative feedback, but now we're going to use the allosteric site in a different way. We're going to introduce a non-competitive inhibitor to bind there, but it still will change the shape. So notice that when the non-competitive inhibitor binds to the allosteric site, the protein folds differently. We've lost the active site. It's certainly changed, and substrate is no longer able to bind, and as a result, no product is synthesized. So both competitive and non-competitive inhibition inhibit the enzyme, no product produced. They're just doing it by slightly different ways. Again, competitive inhibitors compete for the active site. Non-competitive inhibitors are binding to the allosteric site, which in turn changes the shape of the enzyme so substrate can't bind. And again, many drugs that we have either created or discovered act as non-competitive inhibitors to have their effect. A good example is penicillin. So we talked about penicillin briefly way back in our video on bacteria. We were talking about how important the cell wall is for bacteria to protect them. And penicillin, we said, prevents cell wall synthesis. So what's really happening is it's acting as a non-competitive inhibitor for an enzyme that bacteria would normally need to produce the cell wall. So there's an enzyme in bacteria that cranks out the components of the cell wall, and penicillin non-competitively inhibits that particular enzyme. So again, enzymes are needed to catalyze or speed up reactions, but they're supposed to speed up specific chemical reactions. And that's because of the lock and key fit of a particular substrate and the active site. In other words, for every reaction that would need to occur in a cell, there will need to be a uniquely shaped enzyme to catalyze that specific process. So we know that's the job of enzymes, but then we can regulate the enzymes to in turn regulate the amount of product that is being produced. So we're seeing that enzymes play a role in the whole energy harnessing stuff, but we're also starting to talk about regulation, how living things regulate things, in part by regulating their enzymes. All right, so that's what we needed to get into in Energy and Metabolism Part 2. There's plenty more we need to discuss and learn, so we'll get into some more interesting topics in our next video. So we'll see you then.